Welcome to the narrated poster session number 360 of the American Society for Bone and Mineral Research 2022 annual meeting. We are honored to have been selected to present progress on the development of RMD 1101, an FGF18 gene therapy for the treatment of osteoarthritis. OA is a disease that affects roughly 1 in 7 adults and is the leading cause of nearly 2 million hip and knee replacements performed in the US each year. Despite its debilitating burden and the number of therapeutic classes investigated, there are currently no approved disease-modifying treatments. Recombinant human FGF18 analog sprifermin has, however, recently demonstrated remarkable results in a placebo-controlled phase 2 trial, statistically achieving a dose-dependent increase in cartilage thickness, which was coupled with a reduction in the rate of progression to knee replacement and improvement in low back pain in the high-risk subgroup. To overcome the pharmacokinetic challenges which are associated with the protein, Remedium is developing an FGF18 gene therapy which will express the full-length, naturally glycosylated FGF18 locally within the joint. To compare the effects of this treatment with the truncated bacterial analog, we first produced a full-length eukaryotically expressed protein and confirmed its structure and glycosylation. When administered to chondrocytes in culture, both the bacterial truncated protein and the full-length eukaryotic FGF18 elicited dose-dependent chondrocyte proliferation and appeared highly cytocompatible. In parallel, we evaluated the effect of our gene therapy vector on primary human chondrocytes and synoviocytes in culture and confirmed its excellent cytocompatibility and permissivity of both cell types. We saw a rapid increase in GFP-positive chondrocytes and synoviocytes over time, with a clear chondrocyte transducibility advantage in vitro, and we observed no dose-limiting toxicities in either cell type. We subsequently confirmed that the same chondroproliferative effects induced by the recombinant human FGF18 protein can be induced following delivery of AAV2 FGF18 at doses near the bottom of the in vitro confirmed therapeutic safety window, and a dose-dependent effect can clearly be observed with the gene therapy. Using unbiased transcriptomic analysis of chondrocytes treated with AAV2 FGF18 or the FGF18 protein, we observed that both approaches upregulated hyaline cartilage-related genes while downregulating fibrocartilage and osteoarthritis-associated transcripts. Specifically, the gene therapy was able to upregulate Has2, a transmembrane protein responsible for the production of high molecular weight hyaluronan, called 2A1, a chief component of hyaline cartilage, and PRG4, a lubricating hyaline cartilage surface protein. At the same time, gene expression of COL1A1, ADAMS, and LOX, proteins which have been previously associated with scar-like fibrocartilage and osteoarthritis, are suppressed following treatment with AAV2 FGF18. Volcano plot analysis of RNA-seq data further demonstrates the similarity in genes up and down-regulated by protein and gene therapy treatment. Specifically, all genes down-regulated greater than threefold by MOI10 of AAV2 FGF18 were also down-regulated by the high in vitro dose of 1000 nanogram of protein. More detailed analysis demonstrates down-regulation of ANGPTL7, a gene with a suggested role in osteoarthritis, and DSM1, a gene with a previously suggested chondrogenic mechanism. Finally, we analyzed the patterns of gene expression using protein interaction mapping of M-code networks. A total of 9 distinct M-code networks were identified for the protein treatment group, 67% of which were associated with the cell cycle. 13 distinct M-code networks were identified for the gene therapy treatment arm, only 38% of which had a cell cycle association, suggesting a potential benefit of the low-dose gene therapy at increasing mechanistic diversity of upregulated genes with a greater emphasis on ECM production. In summary, the AAV2 vector is highly efficient at transducing primary human chondrocytes and synoviocytes, demonstrating optimal cytocompatibility. Both the gene therapy and FGF18 protein treatments induce similar dose-dependent chondroproliferative activity and upregulate hyaline cartilage ECM-related genes, while downregulating those associated with scar-like fibrocartilage and OA. Both effects are dose-dependent, and relatively low quantities of AAV2 FGF18 can produce comparable biologic responses to protein doses, which previously informed the clinically therapeutic range. Thank you for your time, and a big thank you to Dr. Hollander, Dr. Zhang, and the Zhang Lab members, as well as Dr. Albert Tai of the Tufts University and Tufts Genomic Core facilities.